To all students, I hope you are doing fine. Today in my video we are going to discuss conjugation in bacteria. So first of all we are going to discuss literal meaning of conjugation. The literal meaning of conjugation is temporary union between two cells or individuals. So today we are discussing conjugation in bacteria. So that is the process of temporary union between two bacterial cells. Now let's move on to the definition of conjugation. If we look at the definition of a conjugation, transfer of genetic material, which is usually DNA, or we are going to take an example of plasmid, between bacteria through cell to cell contact. In this process, one bacterium is going to transfer its genetic material in the form of DNA to other cell of the bacterium by cell to cell contact. So let's move on to the explanation of this topic. So I have already made a diagram for you and steps we are going to discuss. So let's move on to the first step of this process. First process will start with the structure of a bacterium. So now we are not going to describe in detail the structure of a bacterium, but we are going to discuss only few things which are responsible for in the process of conjugation. So this is a bacterium. This bacterium is known as F positive. F positive bacterium usually contain two types of chromosome. One is regular chromosome, which is responsible for growth and development, while the other chromosome, other DNA, which is known as plasmid, which is responsible for antibiotic resistance. This plasmid is known as F plasmid, and it is also known as F factor, and F mean fertility, because such bacterium which contain this factor, it has ability to do conjugation. If this factor is not present in any bacterium, then it is not able to do the process of conjugation. So this bacterium which contain F plasmid, known as F factor, is known as F positive bacteria, which is also considered as male bacterium. Remember that sometimes conjugation is also referred to as sexual reproduction, but it is not a sexual reproduction, because in bacteria, in this process, there is no gamete formation and there is no new generation which is produced by conjugation. So it is the only transfer of material, genetic material, from one bacterium to another bacterium. Now the male bacterium, which is F positive, which has F plasmid, has also another important property, that it has a special structure formed on the surface of the body, which is known as sex pilus. With help of this structure, it can contact with another bacterium and transfer its genetic material. Now let's move on to the other bacterium, which does not have F plasmid. It has only single regular chromosome and it does not contain F plasmid. So that bacterium is referred to as F negative, and we will call it female bacterium, and this is usually known as recipient bacterium. So first step, I am going to read for you F plasmid, which contain F factor, which is also known as episome. Episome is a special type of DNA which contain few kilobytes of DNA bases, which is going to be transferred to another bacterium, is known as F positive and also known as male bacterium. And the other bacterium, which is known as F negative, which does not contain F plasmid, which is also known as female, and it is also known as recipient bacterium. They come across each other. Second step, F plasmid contain pylon genes which form sex pili. F plasmid contains special type of genes which are responsible for formation of special structure on the surface of the body with the help of a protein which is known as pylon. This structure is known as sex pilus, which is responsible for the process of conjugation or contact with the female bacterium. Number 3. Polymeric proteins form contact between F positive and negative. When these both bacteria, F positive and negative, come across each other, there is a formation of contact between these two bacteria with the help of sex pilus, and some special type of polymeric proteins helps to attach sex pilus of one bacterium F positive to the F negative bacteria, and then there is a special enzymes which produce a kind of hole between these two bacteria, and these two bacteria join with each other through cytoplasmic bridge. Next step. In the next step, conjugation is initiated by mating signal. As soon as the contact is formed, special type of genes send signals which initiate the process of conjugation. During this process, activation of DNA takes place, and during activation of DNA, certain enzymes which are endonucleases are released, which cut the DNA at a certain point of the plasmid, which is known as origin of transfer. In this red circle, which is a plasmid, you can see the black spot. This black spot is known as origin of transfer. Right here, the enzymes endonucleases will cut the DNA and form a small nick from where the DNA can be separated. So in the next step, you can see relaxosome, which are special type of endonucleases enzymes which are released by cell, create a nick in one plasmid strand. So in the next step, you can see when the nick is created, the double stranded of the plasmid DNA gets separated, and single strand of plasmid DNA is transferred from donor bacterium to recipient bacterium. So if we look at the next step says mating always occur between F positive and F negative bacterium, because F positive and F positive cannot do conjugation, and similarly F negative and F negative cannot do the process of conjugation. So it is always between F positive and F negative. F plasmid, which is fertility plasmid, is transferred as a single strand. So a single strand of F plasmid is transferred from one bacterium to another bacterium. Now these two bacteria, both bacteria, they contain single strand of F plasmid strand. Now they are going to synthesize complementary strand of this F plasmid. Now in the next diagram, you can see that this bacterium has synthesized its complementary strand of plasmid DNA, and its DNA now is complete plasmid, which is known as F positive plasmid. Now this F negative bacterium has been converted into F positive cell, and now this has also ability to do conjugation. In the donor bacterium, because only single strand was left, it is also synthesized by bacterium, and a new complementary strand of DNA is synthesized in this bacterium, and this will again become F positive cell, which is fertility cell. Now these two bacteria can also do reproduction with help of binary fission, and also can do conjugation with F negative bacterium with another type. So this process usually takes in the bacterium which is known as E. coli. E. coli is a bacterium which is present in the gut or digestive tract of human being, which cause infection in the small intestine. So usually these bacterium do conjugation. So in the last step, complementary strands of both F plasmids are synthesized and conjugation is completed. Sometimes some portion of chromosome over here is also transferred from one bacterium to another bacterium, which is integrated into the host or recipient chromosome and become recombination or recombinant DNA or recombinant bacteria. So the question is, why do bacteria do conjugation? So bacteria live in a very competitive environment where they have a tendency to compete with other organisms, and also they have risk of their lives with the help of antibiotics, because human beings use a lot of antibiotics. So some bacteria, they gain a resistance against certain antibiotics, and they want to transfer this technology to other mates or other bacteria. So that is why, to transfer technology or antibiotic resistance, bacteria do conjugation and transfer their genetic material to the other types of bacterium, so that they can also become F positive plasmid or F factors or donor bacteria. So in this video today we had discussed about conjugation in bacteria. I hope it makes sense, and I'll see you in the next lecture. Until then, bye.